The screen is better, so from now on, this lecture will be here, not there. Yeah? Okay, so what do you remember? Sorry, what do you remember? Yes, prokaryote. Yes. Can you give an example of prokaryote? What are prokaryotes? What is the characteristics of prokaryote?
There is no nucleus.
Yes, there is no nucleus. Yes, exactly. They don't have nucleus. Do they have like mitochondria and stuff? Mitochondria. Do they have anything in the cytoplasm? Like, they don't have nucleus, but what about other organisms? What about other things like mitochondria? No, exactly. Well, we will see. We will see. An example of prokaryote. Do you have an example of prokaryote in your mind? Like, this is a prokaryote, right? This is a typical prokaryote. Because you can see there is no nucleus. Inside just looks like some soup. But you can see some dark part and some lighter part. So it's not homogeneous. It's not homogeneous. But what is an example of a prokaryote?
E. coli.
E. coli. Yes, E. coli, dijoking, is a prokaryote, bacteria. But there is, in the prokaryote, there are two big groups. The bacteria, like E. coli, but there is another group that we do not know much about, but they are very important for the environment. What are these? They look a little bit like bacteria, but they are very different. Yes, archaebacteria, yes. These are very, very old, probably the oldest living organisms on Earth. All right. So, we stopped last lecture at the eukaryotic cell. And as you can see, the first point is that the nucleus is the information store of the cell. This is yeast. Okay. You can make bread, you can make alcohol with this, you can make sake, many things. And this is one cell. And we can see inside the cell there is the nucleus. And sometimes this is called budding yeast, because you can see this is the cell and there is a new cell that grows from this one. And if you look a little bit later, it becomes like this, and after some time this will be cut and you have two cells. So this is called the budding yeast. And it is very useful in biology, because what we know about how our cell work was discovered by using yeast. Why do you think, of course yeast has a nucleus, it has almost everything our cell has, but why do you think it is easy to study yeast? Why do you think it is easy even to study bacteria? Many things we know about cells, actually we know because of study in bacteria. Why do you think? Why do you think scientists use these to study eukaryotes? The main characteristic. Why do you think it is important? If you think about yeast.
Yeast does not change genetic information.
Well, that's not exactly true. That's true that yeast is very stable, but yeast will change. Many generations of yeast, they may accumulate mutation and things like this. This is possible. Bacteria, they will also accumulate mutation very quickly. So why are these like bacteria for the prokaryote, and for the eukaryote, why the yeast is the tool that a lot of biology started to use in the beginning, especially in molecular biology. Why this is? Very simple reason. It's a very simple reason.
Very fast to.
Exactly. It is extremely fast. Yeast, if you have a right environment, it will grow very fast. So very quickly you can have many, many cells. Bacteria also. Bacteria, they will divide very quickly. So it is very easy to study them and very cheap. All right. So this is a typical cell, and usually in the very center of the cell you have nucleus. Why the title of this chapter was "This is the information store of the cell"? What does it mean when it says that the nucleus is the information store of the cell? What do you think it means? Yeah, that's the title, right? The title here. The nucleus is the information store of the cell. Informationは情報ですよね. What does it mean? Why do they say that? Can you understand why they say that? Why it is written that it is the information store of the cell?
Nucleus contains DNA, and DNA is the code of the
Genetic code.
Genetic code.
Yes, exactly. The nucleus contains most of the DNA of the cell. The nucleus is just a protective membrane that protects the DNA. So inside the nucleus you have a very high concentration of DNA. And what we can see here, this and this black bits here, this is DNA. This is also DNA, but in a different kind of shape. We will see later what it is. But mainly in the nucleus you have a lot of DNA. All the chromosomes of the cell, they are in the nucleus. Okay? All the chromosomes of the cell, they are in the nucleus. Only one chromosome, which is not really a true chromosome, is outside the nucleus. We will see which chromosome. And if you can see, there is a white membrane here. Actually, it's not a one white membrane. It's actually two very thin membranes that surround the nucleus. You can see here, there is one membrane, and then there is a second membrane outside here. And of course, there are nuclear pores, P-O-R-E, pores. Like maybe we cannot see that, like maybe we can see here, something here, maybe something here. This is holes because you need to have information, like for example, proteins need to go in and out of the nucleus. And also like amino acid, nucleotide, they need to go in and out of the nucleus. So you need to have holes in the nucleus that can be crossed by many things. So it is very important, especially because, okay, now you know there is DNA in the nucleus. So what is one of the very important things that happen in the DNA? The DNA is the genetic information, right? But then we are made of protein, and as I explained last lecture, DNA is used to make RNA. RNA is used to make protein, right? This is very common among many living things. DNA, so DNA to RNA, where this happens? where does transcription happen? Where does transcription happen? You know, you have DNA here, DNA, then it is transcribed into RNA, and then it is translated into protein. Right? So this is in English, this is the transcription. And this is the translation. Okay? The whole thing is identity. This is gene expression. We saw that DNA is in the nucleus. DNA is in the nucleus. So where transcription occurs? In the nucleus, right? So the whole transcription has to happen in the nucleus. Okay. Where does the translation happen? Where the translation happens? I think briefly I said something last week. Where the translation happens.
Ribosome in cytoplasm.
Yes, translation, it's inside, in the cytoplasm with ribosomes. In cytoplasm, right? So this here, this whole thing here, this is actually the cytoplasm. So it is obvious that there need to be some passage because RNA needs to go out of the nucleus to be translated into protein. So this is one of the most important part of this. This is one of the most important part of this nuclear core. It is to let RNA leave the nucleus and be translated into protein. This is very important. Okay? But as we saw, the nuclear membrane is actually composed of two right? Not one single membrane, but a double membrane. So please remember, this is very important. Okay. So yes, nucleus contains DNA, and sometimes with a simple light microscope we can see very easily DNA. This is the nucleus here, and you can see these mushi mitai. This is chromosome, and just before the cell will divide, then all the chromosome will condense. These are condensed chromosomes. They will gather together and they will be very easy to see. So each of these strings, it's DNA. It is chromosome. Okay? It is chromosome. We will see the mechanism of this later. Then a second organelle in the cytoplasm that is very important is called the mitochondria or mitochondria. It really depends. But both are okay. Mitochondria generate usable energy from food to power the cell. By the way, in English, mitochondria is many mitochondria. Okay. How do you say one? Do you know?
Mitochondrion.
Mitochondrion. Yes, it's a little bit difficult, but this comes from the Greek, right? So mitochondria. Mitochondrion. This is hitotsu. This is takusan. Okay. One mitochondrion, many mitochondria. So this has something to do with the energy of the cell. And this is a reconstitution of this one that I showed you before. This one here. And then if you sort of look at mitochondria, this is what you will see. So you have the big cell here and the new cell that just developed from the mother cell here. And then inside, of course, here you will have the nucleus inside. But you can see this network is mitochondria. So in books, usually you have this kind of drawing. This is what you have in book. Everybody, when you ask what is a mitochondria, they will draw something like this, right? Exactly something like this. But one mitochondrion, if you take a look at this, what is one mitochondrion? It is very difficult. So mitochondria is a network. Okay, it's a complete, maybe this is one mitochondria, but one mitochondrion, but actually it soon will move and will be absorbed in the whole network. And it's always moving, always moving. So it is really a complete network inside the cell, like this. But it is very easy to show them like this. And why we show them like this in pictures, it is because if you make electron microscope section into the cell, you will cut this. And when you look at the microscope, you will see something like this. Just because of the way we cut. But actually it's a complete network. This is only a slice of mitochondria, but this mitochondria maybe was very long going that way and very long going that way. But anyway, this is the structure of a mitochondria and of a mitochondrion, if you want. And then you can see here as well that it has one membrane here, and it has also another membrane inside, which is called the inner membrane. It means inside, as opposed to the outer membrane outside. And the inner membrane is very interesting because you can see it sort of makes these many folds like this. As you can see, this is what happens here. And this is very important for the function of the mitochondria. And there is a theory, I really like this because I told you that all the chromosomes, they are in the nucleus. Only one chromosome exists out of the nucleus, and it is in the mitochondria. Mitochondria the 23 class is in 24, especially for today. No, actually from now, because the projector day doesn't work. So
we run up to the first list? Yeah, is that okay?
No, it's all right. Have a seat. Have a seat. We have 2 new students to the lecture, but they could not find because we have moved. So have a seat. So I like these guys because, mitochondria have their own genome. Their own genome. They have one single bit of DNA, which is quite small, only about 16 kilobyte, not kilobyte, kilo base pair, okay? BP, kilo base pair. So very early, scientists were thinking, why mitochondria have their own genome? They are inside the cells. It doesn't make sense, right? The genome is in the nucleus, not in the mitochondria. And when this DNA was discovered in mitochondria, people emitted the theory that actually mitochondria, they were originating from maybe a bacteria that was eaten by an early eukaryote, already with a nucleus that we can see here. It's an early pre-eukaryotic cell with the beginning of a nucleus. And that's still a theory, okay? It was believed that these pre-eukaryotes were predators. They were eating smaller cells. And one of these pre-eukaryotes actually ate a bacteria. And somehow, these bacteria became a kind of, a permanent resident into the pre-eukaryotic cell. And both of them, it's a kind of symbiosis. They live together. Symbiosis, it means living together. And then, this early eukaryotic cell, it was anaerobic. You understand what means anaerobic? Anaerobic. Of course, you guys will know. What means anaerobic? And when you have this prefix, it means no. And aerobic means simply that you are using oxygen. You need oxygen for your metabolism. But if it's anaerobic, it means that you cannot use oxygen. Okay? So this cell could not use oxygen. Oxygen may have been a poison even before. But then from the time it actually engulfed a bacteria, then it gave the ability to actually use oxygen and therefore be more efficient in its metabolism. So this is the theory of the origin of mitochondria. And this event here that, an early pre-eukaryotic cell kind of, engulfing a bacteria that gave rise to this mitochondria later on, it's believed to happen maybe 1.5 billion years ago. And this coincides with a huge surge of oxygen in the atmosphere. So it made sense for cells to actually develop in such a way. Because at that time, the amount of oxygen increased a lot in the atmosphere. And cells needed to use and be protected against oxygen. And as we saw before, so the nucleus is surrounded by two membranes. And the mitochondria as well. So mitochondria have, well, not exactly an outer membrane, but it has a plasma membrane and another membrane at the edge, right? So when this bacteria was eaten by the cell, then the outer membrane of the, there are three membranes, the plasma membrane, the outer membrane, and then the membrane of the cell that ate the bacteria just disappeared. And then this is why these mitochondria still have double membrane that originate from the original double membrane. But that's just the theory. There is no proof of that. There is no proof of that. Only some suggestion. It's almost certain, but there is no definitive proof. It's only a theory. But it's kind of accepted at the moment. Alright. Chloroplast, it's another quite similar organelle. Have you heard about chloroplasts before? No? You have heard about chloroplasts before?
A long time ago.
A long time ago. So chloroplasts, they do not exist in mammalian cells. They are not in animal cells. They only exist in eukaryotic plant. Okay, eukaryotic plant cells. And it looks a little bit similar to a bacteria in that it has, of course, a double membrane. And it also has some sort of complex structure complex membranes within its body, and these are enriched in chlorophyll. This is why they are green. Usually, if you look at a plant cell under the microscope, you will see a lot of green stuff. This will be the chloroplast. So you have a whole cell here, and then you can see the chloroplast. And they are enriched in chlorophyll, which means what is the function of the chloroplast? Why would a plant cell have a chloroplast? Why we do not? What's the function of chloroplast that we don't do? What do you think? Why are plants? By the way, the plants are green because they contain a lot of chlorophyll, which is contained in the chloroplast. But what is the function of chlorophyll?
Generating sugar.
Generating sugar. From what?
From sunlight and water and the
other way around.
CO2.
Yeah, carbon dioxide. Yes, so chlorophyll is actually the pigment that will actually be excited by light and generate the energy to transform carbon dioxide into sugars for the plant. Yes, indeed. And I told you in eukaryotic animal cells, there is one chromosome that is in the mitochondria. If you look at the sequence of the mitochondrial genome, it's actually quite close to the genome of early bacteria, which is another kind of sign that they have a kind of common origin. But in plants, mitochondria also, in plants, chloroplasts also have their own DNA. This is the chloroplast DNA. And so, the same theory that was proposed for eukaryotic cell containing mitochondria was also proposed for plant cells, is that an early eukaryotic cell that already had absorbed its, that already had mitochondria, somehow underwent a symbiosis with a bacterium that was photosynthetic, that could use light to make its own organic molecules, like sugar, that you say. And so this cell, who was forced maybe to eat other cells to survive, suddenly became able to just synthesize sugar and many other things from light, just because of this symbiosis between a photosynthetic bacterium and the eukaryotic cell. And then, in time, this became chloroplasts. Okay. Again, that's a theory. But there is some evidence, like, for example, as I said, the genome of the chloroplast is quite close to the genome of photosynthetic bacteria, some of them at least. So there is kind of good evidence that this is really what happened. Okay, so we saw the nucleus with double membrane. We saw the mitochondria with double membranes. We saw the chloroplast with double membranes. Membranes are important. If you have to remember something, is that your cell is mostly made of membranes. Okay, they separate many different compartments with very different functions, and this is the next point of the lecture. So we already saw this nucleus. We already saw the mitochondria, and then you also have this huge network which is connected to the nuclear membrane, and that is here pictured as electron microscope picture there, and you can see all these membranes here, which is represented here as a cartoon. And as we saw last week, all these single dots, all these black dots, they are really huge macromolecules that are, what are these guys?
Ribosome.
They are ribosomes. They are ribosomes, yes. So as we saw in this picture, that you have DNA that is transcribed into RNA, RNA is translated into protein, translation, this is where it happens. This is where all the ribosomes are located, and RNA somehow are carried there, and they are translated into protein. And there is another organelle, okay, another subcellular compartment, which is called the Golgi apparatus. And this is a very important, it's like tacubin. It's like the post of the cell, okay. Everything that is produced by this structure, by the endoplasmic reticulum, somehow is carried into this Golgi apparatus, and then you can see these little vesicles here. They bud out, and then they sort of merge with this Golgi apparatus, and then vesicles will leave the Golgi and carry these substances. It can be many things. It can be protein, it can be fatty acid, many things. Will then go where they are supposed to go. We don't really understand at the molecular level how all this trafficking happens, but it does happen. So, for example, most of the fatty acids that are component of the membrane, they are made in the Golgi. Okay. So, this is, remember, this is kind of a transport structure, and the next is this kind of general view of a cell. So we saw the nucleus, we saw the endoplasmic reticulum. We will see the function of these in more details later. This is just the introduction. We have the Golgi. Peroxisome and lysosome, they are a little bit different. They are also very specialized in their own function. Okay. The lysosome, this is the stomach of the cell. The image, okay? This is where most of the food that the cell receives will actually be chemically digested, okay? And then maybe free amino acid, free nucleotides will actually be released from the lysosome and will be recycled or further broken down. The peroxisome, they are also very specialized, and this is where, do you know hydrogen peroxide? Hydrogen peroxide? This is actually a poison. Okay. This is almost like water, but it has 2 oxygen. It's called hydrogen peroxide, and it's extremely reactive molecule. Extremely reactive molecule. But in our metabolism, we need small amount of hydrogen peroxide because there are some chemical reactions that require hydrogen peroxide. Notably, the detoxification of substance need hydrogen peroxide. So peroxisomes, they are very, very specialized organelles that do these reactions. Okay. They contain a lot of hydrogen peroxide inside. This is why they need to be protected from this is why the cell needs to be protected from this. So that's the function of the peroxisome, is to carry out hydrogen peroxide and related functions. Therefore, it's called peroxisome. And then, all these membrane compartments, all these membranes, you remove them. Okay? The whole thing, this is the nucleus, and we saw that this is called the cytoplasm. Okay? But if you remove all this structure, and you're only left with the liquid inside, this has another name, which is just the cytosol. Okay? This is the name of the cytoplasm without the organelle, without the membrane-limited compartment. Okay, so I told you about this Golgi apparatus, but actually, if you could look at a live cell, it is possible to do. Always the cell will take up some food or substrate or whatever, and then this will be carried out by vesicles where they are supposed to be carried out, and then they will be metabolized, transformed, whatever. And then after that, the cell will generate some, some whatever substance, either like garbage or like some things that need to be exported, like for example some hormones, for example, and then they need to be exported out of the cell, and this is using the same kind of road. So things are absorbed by the cell by doing this kind of endocytosis, endocytosis, and then carried into the cell, and then cell gets rid of its content by exocytosis. Okay, these are the two important roads for the cell. The cytosol is a concentrated aqueous gel of large and small molecules. Yes, so I told you, if you remove this structure, this compartment, you are left with a liquid, but that's just not water. It is highly, highly concentrated in many things, especially proteins. Especially the cytosol contains a lot of protein and a lot of, mainly protein. So you can see this doesn't really tell you much, but this is the cell membrane here, and you have many kind of strange arborescent things. But this represents to you how crowded it is inside the cell. It is just not a bag full of water. It contains a lot of things, and everything is organized, and everything is where it's supposed to be. Okay. Well, there is a randomness into it, but as you can see, it's very organized. So you have these strands. These are proteins of the cytoskeleton. Then you have, maybe this might be the nucleus here. What could be this thing? Do you recognize?
Mitochondria.
That could be mitochondria. We can see the kind of sign of the double membrane there. Yeah, so there are many, many things. Okay, it's a very, very crowded mix of molecules. Some big macromolecules and some very small molecules like nucleotides or amino acids or even salts, water molecules also. Okay. So these things which I just told you about, these strands of stuff, this is called the cytoskeleton. And the cytoskeleton is responsible for directed cell movement. It's just not a cytoskeleton. You know, cytoskeleton, you sort of have an idea of something that is rigid and doesn't move. But actually this is not true, as we will see later. So there are mainly 3 different types of macromolecules in the cytoskeleton. You have actin filaments, and actin filaments, as you can see, they really crisscross the cell to give it rigidity and structure. You have the microtubules. They are very different because microtubules, they really surround the nucleus here, and they also connect the cell membrane with the nucleus. And microtubules are also very important for cell division. When a cell divides, all this reorganization of the cell is mediated by the microtubules. And then you have the intermediate filaments. You can see here. It's another type of protein. We will see again what these are made of. But that's another type of protein. These are completely different molecules. This is actin. This is the name of the protein. Microtubules, they are made of tubulin. This is another protein. And intermediate filament is yet another protein. But if you look at this like this, it's just a fluorescent microscope picture. You have no idea of how dynamic it all is. But if you look at a cell dividing, for example, so this is a separation between a future cell and another future cell. And they both come from the same original mother cell. And each of these black things here, this is a chromosome. This is a chromosome. And each of these chromosomes will actually be pulled apart. So this chromosome will be pulled here, and this chromosome will be pulled down there. We can see this network of microtubules here. These microtubules will be the structure that will really pull the chromosome apart into each future cell. So this is why I said that microtubules, you can find them in a normal cell, but in the dividing cells, the distribution of microtubules will be a little bit different. and they will be very important for the separation of the chromosomes. But we will see. This is just the introduction of this. And then, this is great. It's just picture, but if you actually look at a cell, this is a film. This is a film in real time. It's not in real time, it's accelerated, but this is different cells in culture. And you can see cells move. If you look at them under the microscope, even if they are alive, you will not see them moving because they are very slow. But if you have a time lapse like that, maybe you take one picture every 10 minutes and then you sort of make a film, then this is what happens. They are always moving. And all these cellular movements, they are mediated by the cytoskeleton. They are mediated by the cytoskeleton. And then you can see that the front and the back of the cell is also very different. Because cells do have a polarity. When you look at the microscope, where is the front of the cell, where is the back of the cell is very difficult. But actually cells do have a polarity, which is very important. Okay. And this one is a different cell. This one is a protozoa. It's called an amoeba. Maybe you have already heard about amoeba, but this is a single cell organism. It's an eukaryote, and we can see the nucleus here. And this is a huge cell that can actually eat other cells. It's a predator. And then as you can see, it also moves. And this movement is also mediated by the cytoskeleton. And inside we can see some things being moved around. These organelles being moved around. And this all happens because of the cytoskeleton. This is a very thin section in a plant structure, but it is also a time-lapse film. And we have one cell here, and we can see each chloroplast. And then if you actually make a film of this, we can also see this chloroplast moving around. Mitochondria, as I told you, they also move around. Some of them can really go very fast from one end of the cell to the other, especially for neurons. Some mitochondria can go from the cell body down to the axon at quite high speed. Why? Why? The cell moving, you understand. The cell wants to go where it wants to go. Fine. But why would the cell, especially this is in a stem, the plant, they really do not move much. They do move, but they do not move much. But why would the inside of the cell, why would chloroplasts move around? What would be the reason for that? Why would mitochondria move around? Why do you think this?
Chloroplasts should catch light or escape from light, strong light.
That's very interesting. That's a very, very nice comment. Actually, it's true that the chloroplasts will move to catch actually as much light as possible. They want to receive light because they want to, make sugar. It really depends at what time of the day. But if you actually look at this, if this was a film across 24 hours, we would see chloroplast moving from one end of the cell to the other. So if light is on the left, all the chloroplast will go on the left. And if light goes on the right, then the chloroplast will move on the right. So they really follow the light. And even if you switch off completely the light, there is no more light or there is no more darkness. It's a constant environment. Still, the chloroplast will move. Why would that? So, if you put the plant into a light-dark cycle, the chloroplast will sort of follow the light. Okay. But if you give the plant a constant environment, usually in plants, constant light is used, not constant darkness. If you give the plant constant light, so light is always the same, always the same amount of light, always the same, lux and whatever. And still these chloroplasts are moving. What could this mean? What could this mean? I can actually show you one. It's from my next lecture actually, but it's appropriate here. This is. I think it's a beautiful picture. It's not quite a film, but this is taken from a very recent paper, I think in Science. This is a young sunflower plant. And here, this is the east. Where I am is the east, and the sun will rise and set in the west. And then you can take a look. We will not see the sun, though. You can sort of know where the sun is because the plant will really track the sun during the day. And then at night, which is probably now, then the sunflower plant will just go back to where it was in the beginning, so that before the next day, it's already at the right position to receive maximum illumination on its leaf. So this also happens inside the cell at the chloroplast level. Okay. And something great is that even if you put the plant in a constant light, still it will do that. Still it will go like this. And still the chloroplast will move around. So what does it mean? What could this mean? What could this mean? Don't hesitate. What could this mean? You want to say something, I can see.
Me?
No, her. Or you can say something if you want. What could this mean? So, if the sun is there and rises and then sets, that's fine. The plant is following the sun. That's just a response to the sun. But no, I told you that actually if you just put this plant into a room with a constant light, still it will do that. Even without the sun changing position. So what could this mean? You need more experiment than that, of course, to confirm, but it's an indication of what? It's the same with the mouse. It's the same with the mouse. I don't think nobody has studied actually. I tried, but I didn't manage. But like, if the mitochondria also change according to the time of day, there is a lot of variation. So maybe they do not. But if you like that, if you put a mouse in a cage that has a running wheel. This is not in the book, by the way, it's an extra. If you put the mouse in a running wheel and you connect the running wheel to a computer so you know when the mouse is running, and then you put the mouse in a room where 12 hours of light are followed by 12 hours of darkness every day after day after day after day. And then you can see that the mouse will start its activity just at the onset of darkness. That's normal, the mouse is nocturnal. So the mouse will start to be active in the dark. If you put a human into the running wheel, the same thing happens. But it's opposite. Humans are diurnal. So they will be active during the day. But the great stuff is that if you switch off the light completely, because when using nocturnal animals, you use constant darkness, not constant light. There is no more light. It's always dark for weeks, months, but still the mouse will be pretty happy to continue with the same rhythm. So that's the same stuff. You know, this story with the chloroplast moving around, changing with light, but they will keep the same movement even when there is no difference in light. So what does it mean? What does it mean? I will try to show you this chloroplast changing side of the leaf. Actually, during the night they are under the leaf, during the day they are on top of the leaves to get as much light as possible. Or even when the light doesn't change, still the chloroplast will do like this with 24-hour rhythm. So what does it mean?
They have something like
Yes, they have something like rhythms. Yes. But you need to be more precise. What do you mean behind the word rhythms? Because rhythm is many things. In a light-dark cycle and in a constant environment, they have rhythms.
24-hour cycle.
Yeah, 24-hour cycle rhythms. It doesn't change much if you have a light-dark cycle or if you have a constant environment. You will see about 24-hour cycle. What does it mean? This is what you want to say with they have rhythms, but you need to use the right words. What about you guys? What do you think it means? And this is why you are suffering much when you go abroad. Because, if you take the airplane and then suddenly your body thinks it's like 3 o'clock in the morning and then you see a shining sun, that's going to do things to your organism. So this is exactly the same. That's the same reason. That means that we have a clock. All these things are designed to, of course, respond to the environment, but they're also designed to anticipate changes in environment. Okay? So even without changes in the environment, we have the same rhythms because they are endogenous. They are encoded in our genes. This is the biological clock. This is the circadian clock. Okay? So you imagine a plant who has to put its chloroplast when the sun shines. And then you compare two situations. One situation is a plant without clock. Only the plant responds passively to light. Then the plant will be there with its chloroplast at the bottom. And then only when the sun rises, maybe after 2-3 hours, right, there is light now, so I'm going to put my chloroplast up. And then it will be efficient. But now you have a plant that kind of predicts when this light will happen. And when the sun is just about to rise, the chloroplast already go up. So that when the sun just goes up, then already the plant is more efficient at capturing light. So this is the anticipatory mechanisms by the clock. So it's a little bit difficult discussion. But, so all these things, not only it's a response to the environment, but it's also controlled endogenously. And this is a film I actually made several years ago. Here you have cells with a circadian clock, whatever the thing. So these are two different types of cells, and I made a scratch between them. So this is a different cell type from this one. And then here you can see there is no cell. What I wanted to do is there any difference in the speed of movement between these cells compared to this one? And as you can see, these cells are very, very fast. And we can actually see them dividing in real time. So this, for example, is a cell that just divided this one as well. As you can see, these cells are very, very quick at moving down, and these cells, they seem very sluggish, and also how they look is very different. These cells are very big with a huge cytoplasm. These cells are very small and kind of a little bit aggressive. And then the two will sort of confront each other. And you can see they sort of have a reaction when they go down there, they have to go up a little bit because maybe they see something that, well, it's not like what I'm... Anyway. So all this movement, this is the cytoskeleton. Okay. At the molecular level, all this cytoskeleton is always being rearranged and always making sure that the cell is mobile. All right. Okay. So the next point is the cytoplasm is far from static. This is what we just saw. But the bacteria is exactly the same. In bacteria, there is nothing much that you can see under the microscope, but still a bacteria will have a lot of molecular movement inside its cytoplasm. But all this thing that I just introduced to you over the last few lectures, it's accumulated very, very slowly, especially in the beginning. If we go back to 1665, this is where Hooke, the guy who really the first one to actually make a very primitive microscope, he is the one who actually created the word cell, as we saw before, because he thought it looked like the monk cell, okay, in a church or something. Then in 1674, Leeuwenhoek, he also used this similar type of microscope, but he discovered protozoa, amoeba and things like this. So you can see, this is in your book. So you have a lot of different discoveries, like Fleming in 1879, he actually, this chromosome described with great clarity, doesn't really mean much, but describes the great clarity chromosome behavior during mitosis in animal cells. So what this means is what happens to the chromosome during the cell division. Is the person actually, for the first time, using a microscope, actually reported that very nicely. What happens to these chromosomes? Where do they go? Things like that. So, the Golgi, which I told you about, was discovered in 1898. Wow, you don't need to remember all these things. This is just factoids, right? But like in 65, that was kind of pretty important. This Christian de Duve is a Belgian guy. He and his colleague, for the first time they sort of fractionated the cells into different subfractions. One containing the peroxisome, the mitochondria, lysosome. Because to know the function, you need to separate them. If you cannot separate them, you don't know their function. So this was actually very important to know what was the function of each of these. And this is only in 1965. It's not so long ago that the function of mitochondria, peroxisome, and lysosome were discovered. So not so long ago. But from there, this is more or less the, where molecular biology started, and then it became much, much faster at that time. Okay. So that's the summary of the introductory chapter. So you have a very nice picture of a plant cell, a picture of an animal, I'm talking very an animal cell, a plant cell, and a bacteria. And they all have the nucleus. The animal cell and the plant cell, they have the nucleus, but this does not exist in the prokaryote. Mitochondria in both the animal and the plant cell. Chloroplast only in the plant cell, but no mitochondria, no chloroplast in the prokaryote. Cytoskeleton in animal cell and the plant cell. Cell membrane, everything, they are both in the plant and the animal cells. But as you can see here, there is no, inside the cytoplasm, you cannot see any membrane. It's just like a mix of molecules. And something that is important in the plant cell is this very big central vacuole. This is filled with liquid and under pressure. Why? So you have also something that is linked to this vacuole is this very thick cell wall of the plant. In plant, this is made of cellulose. It's quite strong polymer, okay? It's a protein, and it's very thick and very rigid. The cell wall also exists in bacteria, although it's made of a different molecule, molecules. But why do you think this vacuole is under pressure? I told you, so you have a cell wall that is very rigid, and then you have this vacuole which is under pressure. So if you put like something that has pressure inside a limited compartment, what's the mechanical consequence of that? This mammalian cell doesn't have cell wall. It's kind of freely moving. Well, unless it's part of a tissue, but even in tissues, animal cells do not have cell wall. They are just limited by other cells. But in a plant, every single cell is encased in a very rigid cellulose cell wall. Why do you think it's so? And it's filled with water, with some pressure. Why this? Can you guess what's the mechanical property of that? Think of a plant, especially a new sapling or something, or a flower, a long flower that's perfectly straight. Even when there is wind, it sort of goes a little bit like this, but it doesn't break. It's just peaking. What happened? What happened? Think. You have something. You have a box, a very solid box, like a cardboard box, and then inside you put something that has pressure, and you try to fit it into the box. And the box has to be strong enough, right? The box has to be strong enough. But then, what properties will this give to the box? What property this will give to the cell to have this high pressure balloon inside the cell, limited by a very rigid cell wall? What will happen? Well, nothing happens, but what's the consequence? What's the consequence? It's not really biology, it's more like physics. What about you guys? No?
Maintain the shape.
Yeah, just maintain the shape. It will be very solid. Why plants are able to stand quite tall? Just think of a bamboo or something. Of course, bamboo has many other things, that make it stand up. But essentially, this makes the cell very solid, very rigid. And when the plant lacks water, if you don't water your plant, then the water content into the vacuole will decrease, and the cell will become much less rigid. This is why plants go like this when you don't give them water. So that's very important for the shape of the plant. All right. So this is what I told you earlier. It's kind of a synthesis of what I told you. So this is more like the origin of the cell. And you have the ancestral prokaryote, right? The very first cell who gave rise in the course of evolution to all these different cells. So the ancestral prokaryote first sort of evolved into three branches. One, the bacteria, then the anaerobic ancestral eukaryote, and then a completely different branch, the archaea, which is actually now, strangely, in the same type of cell family as the bacteria. So these are the prokaryotes, archaea and bacteria. But then when bacteria sort of developed, then some of them acquired some very specific properties, and at some point they form a symbiosis between one of these anaerobic ancestral eukaryotes, and this gave rise to 3 branches that in the future gave plants, animals, and fungi. And then one branch here of bacteria acquired the ability to photosynthesize, but then at some point during evolution, some of these bacteria, they had a symbiosis with some of these cells that already had acquired mitochondria, and this gave rise to plants. That's just a history as we kind of understand it at the moment. But again, there is no really solid proof. This is only based on what we observe, but no definitive proof. So now you have then the non-photosynthetic bacteria, you have the photosynthetic bacteria, plants, animals, fungi, and archaea, and these are the different types of cells. All right. So this is about 1.5 billion years, so quite some time. And why we think these things happen, this ancestral eukaryote could sort of eat, and get bacteria and become mitochondria and get chloroplast and become plant, is because if you look at most of the single cell eukaryotes, most of them are actually predators. Like this guy, it's called Didinium. The name is not really well shown here. It's called Didinium, and this guy is just a predator. It will swim around. You can see it has a lot of, flagella here, and then it can actually swim very quickly. And this is actually technically the mouth of these organisms. It can actually eat other types of cells. Like for example, this guy is now eating another protozoa smaller than itself. So most of these single cell eukaryotes are predators. So this is another kind of maybe observations that somehow most of these ancestral eukaryotes were also predators, and they spend their time eating, engulfing other cells, and just at random at some point then there was a symbiosis being created by these events. But we don't really know how it happens. No, we have no idea. But this is just, as I say, in theory. And well, the shape and function of all these protozoa, these, unicellular eukaryotes, there are a lot of them. A, C, G, A, C, G. These are called ciliates because they have a lot of little hair around their body. Then B, you have heliozoan. Actually, they are called sun animals because if you look at them under the microscope, they really look like suns. They are really bright and they do very funny things with light. So that's really nice. Well, whatever the names, but these are different types of protozoa, and most of them are predators. They will eat other animals to survive. So they don't really photosynthesize. This guy actually photosynthesizes. And this is the one we have here. So that's his name, right? This is a Nuglina here. And then you can see it's swimming around. It's a very strange way of locomotion, but that's how it does. So not only it's a predator, but it can also photosynthesize. And so of course it's a predator, so it's able to move around. Okay. All right. And I think that's the end of today. We saw quite a lot. So the next time we will go about the model organisms that are used in biology to understand how we work. Plants, Drosophila, budding yeast, brewer's yeast, whatever. Okay, this is what we're going to see next time. Okay, this one, for example, it's a plant. Do you know the name? No? The most common plant studied in biology. Maybe you have seen in the newspaper or something. This is a weed. This grows everywhere. Therefore, it's very useful for biology because it grows very quickly. It makes a lot of seeds very quickly, and it's easy to grow. This is In English? Arabidopsis. It's Arabidopsis thaliana, one of the very first model in molecular biology. So anyway, we are going to see that next time. So if you have any question, don't hesitate. What about the speed? Is it quick? How you speed? No. A bit too fast? No. Is it too fast or not? What do you think? ちょっと早い? It's okay? No?
A bit fast.
A bit fast? Okay, so I will. Yeah, maybe I was a little bit rambling today. Sorry about that. I will control my level and make it easier to understand. Do you have any question? What we said today? It's just introduction. That will give you a kind of idea of the type of things we are going to see in this lecture. No? Okay, then you can go. No problem. It's time. So, what about you guys? Where do you come from? We did that, all of us, last week. Where do you come from? What do you study? And what do you want to learn?
What do I study? I come from New Zealand.
Yeah.
What do I study? I study both engineering and economics.
Engineering and economics.
Yep.
Right.
But not biological, like not biomed.
Okay.
Yeah.
Okay.
What I hope to learn? For me, just some revision about biology, you know.
I see some basic stuff. Nice. Okay. So I hope it will correspond to what you want. What about you?
I'm from Finland.
Mm.
I study Asian studies.
Asian studies.
Mostly focused on economic side. Yeah. Well, it's kind of interesting, and of course I studied all this, like the basic biology stuff in high school in Finnish. So
Right.
it's a bit difficult with the terms and stuff. But I'm not sure. Maybe I can mention.
Okay. Yeah, so then it will also be useful for you if I go a little bit slower and maybe write the terms.
Mm-hmm.
Okay. So the book that I am using, I will not see all chapters because we only have 14 lectures, so I cannot see 14 lectures, see this book in 14 lectures. But that's the book I am using, which you can find in most libraries. It's a little bit expensive. It's like 1万円ぐらい, 1万5千円ぐらい. But you can make copies in most of the libraries in the university. And I more or less follow this book, which I find the English is really nice and very elegant English, so I recommend this book if you want to see. This is mostly cell biology, so there will be no physiology or things like this. Just, if there is physiology, this is cell physiology. You won't have more information about like the relationship between the brain and the liver and these things. You don't find that. Just basic cell. Okay, that's it. Can see you next time. Yes, question. Nice, I like questions.
I was absent from this
Yes.
class last week.
Yes, it's your first time. So tell us.
I'm Shibata Mei.
Mm-hmm.
I major in agricultural economics.
Oh, I see. And so you have very basic knowledge of biology, or almost nothing more. At school, did you learn biology? At school? Same thing. 学校です。So, okay. What do you hope to learn? 何を勉強した？言葉か。言葉ですか？生物学はまあまあけど、言葉は一番。Yeah, so there is a mix. There is a mix of people who want to learn biology, and there is a mix of people who want to learn English. So unfortunately, I'm from Belgium, so I am not sure I will be able to implicate. very good English. But anyway, yeah. I mean, the world is large. Okay, so if you have any questions, don't hesitate. It's the end of the lecture for today. Thank you very much. Do you have access to classes?
Yes.
So I put the slides, I uploaded the slides last week of this chapter.
We have to change our courses and the adjustment period now because so we registered for like a set of courses and then it's closed now.
Right.
So they told us to come back during adjustment period.
Okay.
So I think once that's done, we will have access to those courses.
All right. Okay. So without that, you don't have access to this course. All right. So maybe I can make copies for next week of this. That will be possible. Okay. Thank you. クラシズは大丈夫ね。スライドは。どんどん落ちてください。全部。おお、勝った。あ、そうそう、はいはい。今のyes. So now I saw everything. So you can.
スライド、あのスライドっていうのは、
あのスライドは
あれってエッセンシャルバイオロジーから取ってきたやつなんですか？
それは分からない。
チャプター1、チャプター1、チャプター2、チャプター3、チャプター4、チャプター5、チャプター6。チャプター6は分からない。it depends.
そうなんですかね。いや、僕よく分からないんですが。
Sometimes I don't have time to see.
何が分からない。
いや、つまりそのスライドを印刷して持ってるだけでいいのか、それとも本をやっぱり借りてきて送りまで。
多分スライドだけでも大丈夫なんじゃないかなと。
Yes. But it is less expensive than paper. けど時間はけどまあ yes, I think if you copy it is maybe you can copy one page. It's only 70, 80.
What kind of exams going to be like? I'm just like a bit afraid, like if I can.
No, you should not be afraid. I don't think you should be afraid because most of the people are in your case. So you really have. You should not be afraid. And the exam is going to be for this lecture. It's just like a multiple choice question.
So you don't need to actually memorize all terms.
You don't need to memorize the terms, but you need to know what they mean. Like, you don't know what the mitochondria is.
I think like for example, if you're supposed to write an essay about like some kind of
No, they won't be. It's the question I'm trying to give students. It's more like, of course, there will be A, B, C questions, but these are not really interesting. It's more like you have a text and you look at the possibilities and you need to make conclusions. But they will be easy. Maybe you need to write small paragraphs that they will know because.
Like, for some reason, I don't know, in Finland, like, high school, the terms we use are like, they're not close to like English terms. And I'm like why? Like, if they had used like something like close to English, it would be really much
Plus it's an Indo-European language, right? It's an Indo-European language.
Yeah, but it's still like pretty far away from other.
Really? That's interesting.
I don't know, they use some words.
You'll be okay. Don't worry. Don't worry. And if you can get the book and read it in the library or something, that's I really Thank you very much.
this slide is this slide
In the book?
in the book?
ありますよ。ありますよ。これです。チャプター1。ね、これほらほら。同じですよ、ほら。これはこれは inside the book ね。This also, this one also. So all the figures I show, they are in the book. This is in the book. So everything is in the book, almost everything. ね、I show this one ね、先週。これも今回はね、今回。これも今回、これも。All figures I show, all figures. これもね、ミトコンドリアの話、クロープラスの話。これもね。全部あります。
What type is the examination?
It's a good question. The examination is, I can show you actually. The examination is like this. The examination is like this. これは去年のこれだ。Indicate whether the statements below are true or false. If a statement is false, explain why it is false. A. The chemistry of life is carried out and coordinated primarily by the action of small molecules. Carbon-based compound. それだけ。で、どっちが嘘つきか、どっちが正解か、このタイプ question で、which subatomic particles contribute to the atomic mass for any given element? Protons and neutrons, protons and electrons. This kind of questions. This is chapter 2. Chapter 2の質問。だからまだわからないかもしれないけど、this kind of question. And 細胞の話は、これは chapter 1